Look, good afternoon, everyone, and thanks for joining us. Welcome to the ASBP's second PlantChat webinar. And to open, I'd like to acknowledge and pay my respect to the traditional custodians of the land, the unceded land on which we in Canberra are meeting, the Ngunnawal people, and pay my respects to elders past and present. We'd like to extend that respect to all First Nations people as the traditional owners and custodians of this country and honour their cultural and spiritual connection, their continuing care and deep knowledge of country. I want to further acknowledge all First Nations people joining us today. I'd like to make a little bit of an observation about the rich tapestry of Indigenous plant knowledge and how that's woven into care of country. But it's perhaps not well understood by most Australians. We all know that First Nations people have used plants over thousands of generations for nutrition, medicine and technology, as well as for shelter, and rely on plants as important seasonal indicators. First Nations peoples have taught us that plants are our living signposts of what's happening in nature, especially with the climate and around the topic we'll be talking about today, which is disease. Now, before I introduce Bob, I just want to tell you a little bit about the Australian Seed Bank Partnership. We're a 20-year-strong national collaboration which involves 11 conservation seed banks at our state and territory botanic gardens and the key non-government plant conservation organisations around Australia, as well as the Millennium Seed Bank in the UK. Now, the focus of our partners' work is to secure seeds and other plant material, primarily those species that we're concerned about for conservation. And we do this for two key reasons. We want to provide insurance against the loss of our native plants, and we want to ensure genetic material is available for plant recovery, our biological research and conservation, including genetic analyses for effective restoration, climate and disease resilience, which includes Myrtle Rust, and Bob will touch on some of that later on. Now, our partners conduct germination trials on the collected seed and plants, so we know how to germinate them. Some seed is grown on in our living collections, which provides additional insurance, and some is propagated for translocation or restoration. Now, our NGO partners, who Bob is here today as the outreach delegate for the Australian Network for Plant Conservation, have led on best practice in Australia, developing guidelines about how we collect germplasm, how we translocate, and they undertake a lot of national advocacy, and that's particularly been the case with Myrtle Rust. Importantly for today's talk, the network has driven the work to assess the threats of Myrtle Rust, to track and monitor spread, and with our partner botanic gardens and state government agencies, as well as Indigenous and private land managers, looking at the science we need to inform recovery action. Now, Brad's going to put some links into the chat for some of the key documents and some of the research that's been going on in the Myrtle Rust space. Now, about Bob. So I just referred to the work of the network for plant conservation, and Bob has driven most of the foundational work on Myrtle Rust and developed the National Action Plan with partners, including Dr. Jeff Pegg and other Myrtle Rust researchers. He is actually what I call Australia's Tsar of Myrtle Rust. Now, we attached Bob's bio with the invitation. He's an absolute giant of the plant world. And while we know you'd like to re-immerse yourself back in the world of taxonomy, we really need to get that national coordination and investment going on the Myrtle Rust, in the Myrtle Rust space. Now, Bob is going to have some practical and cost-effective ideas and talk to you about recovery today. That will build on the learnings to date from the active Myrtle Rust recovery network that's in place. A lot of you are online. We really need to be raising awareness around the work of this space, and this webinar is one way to do that. So I will now hand you over to Bob, and we have a Q&A function in the webinar at the top of your screen. So please place your questions in the chat, and Brad will kind of moderate that, and we will get to them after Bob's presentation. So over to you, Bob.
Thank you, Kathy. Am I coming through on audio okay?
You are.
Right. My name is Bob Makinson. I'm a conservation botanist based in Sydney, and Kathy's right, I was a happy plant taxonomist until Myrtle Rust came along. Those were the days. I would like to acknowledge also the traditional owners, past, present and emerging, of all the countries that we're meeting on today. I'm on Darramurrugal land in northern Sydney. Like all the conservation work that we're all involved in, Myrtle Rust issues really bring to the forefront the need to think about what custodianship actually means in the context in which we find ourselves today. So I think we all need to chew on that a lot more than perhaps we do in ordinary day-to-day life. Now, there's been a certain amount of mission creep in this talk. It was advertised as recovery strategies for Myrtle Rust species, and I was going to talk just about problems of germplasm capture and maintenance and use in that context. But it has sort of blown out a bit, and I really want to talk about the looming, or what I see as looming bottlenecks in ex situ conservation, and why we need to think bigger, but with examples from Myrtle Rust and from a couple of other trending priorities as well. And just as disclaimers, this is a presentation of personal views, not necessarily those of ANPC or anybody else. And I do acknowledge that my approach, just because of my situation these days, is East Coast centric. The bottleneck factors to worry about in WA, for example, are, as I understand it, rather different. But in Eastern Australia, I think we do face some coming bottlenecks, and I want to try and outline my thinking on those. Now, I'm assuming that everybody here today knows something about Myrtle Rust, and I won't dwell on the introductory stuff, other than it's an exotic disease first detected in 2010, just north of Sydney. Within 2 or 3 years, it was up and down the entire east coast. It only affects plants in the Myrtaceae family. Its habitat envelope is basically fairly moist environments, so it's not a problem in the inland. It has a huge host range, over 250 native Australian plants so far, and that could well increase drastically if it ever gets to southwest WA. We don't know quite how it's going to behave if and when it does get there. Out of those 250 odd Australian hosts, about 20 species are in severe to catastrophic decline as a result of this one pathogen, and there's another 20 or 30 or so on the slide as well. One of the catastrophic decline species that's been getting a lot of attention is native guava, Rhodomyrtus pseudioides. Before 2010 it was common and widespread. It's now critically endangered. And it's been reduced basically over virtually the entirety of its range to these fields of suckers that you can see coming up in the shot at left there on the surviving root systems of dead adult plants, putting on just enough photosynthesis to keep going from year to year, although for how much longer we don't know. Now, that species, native guava, and a number of others in this crash decline situation, we really don't have much in the way of alternatives for how to think about recovery processes. There's no treatment available in the wild. There are no refugia to speak of within the natural range of these species from the Myrtle Rust pathogen, and resistant genotypes are rare. They probably do exist, although not necessarily at a very strongly exhibited level. And really we're looking necessarily at an ex situ conservation strategy of great urgency if these species are to be saved from going completely extinct. And the vision for species recovery that's outlined in the National Action Plan and that has been followed in New South Wales, and I think is being taken up in Queensland now as well, is to secure the wild germplasm as a matter of absolute priority on as broad a basis as we can, so that we're capturing the natural range of genetic variation within the species, and to get that into protective custody, if you like, in a situation where we can identify any potential resistant genotypes, or at least genes, which can then be selected for and bred up and back-crossed eventually into, by classical means, not by genetic engineering, back crossed into the wild genotypes so that we wind up with something that could be reintroduced into the wild that is as close as possible to the regional variant that it originally came from, but with the addition of a greater degree of tolerance of the Myrtle Rust pathogen, not likely to be absolute immunity. So that's the kind of schematic of the recovery strategy for these crash decline species, and that big blob in the middle of safe custody involves a lot of things and requires a lot of prerequisites. Now, to continue the native guava as an example, it produces these wonderful sucker units still, thank God. We just about would have lost the species altogether if it hadn't done that. And you can also strike from normal aerial shoot cuttings as well off those suckers. It's quite precocious. It'll flower and fruit within a couple of years in cultivation. There's no seed being produced in the wild, so we're dependent on ex situ work to produce seed for both seed storage research and general seed biology research. And there's some tissue culture work going on as well at the Australian Botanic Gardens, Mount Annan. So the next steps with this species are to screen for rust resistance, and we expect that to be quite difficult and prolonged process because there's no exhibited resistance in the wild that we are aware of, unlike some of the other crash decline species where there is. The trick will then be to identify that low level of incipient resistance and select and amplify it, and then conduct field trials and take it from there. Now, native guava goes like a bomb in protected cultivation, which is a good thing, yeah? It's got to be good. Means we can bulk it up rapidly, we can develop a seed production capability quite rapidly, and we can produce it for resistance screening arrays, which can be quite large, although that's not yet been commenced for this particular species. But its amenability to cultivation is also not good because it means frequent repropagation of potted plants. A lot of nursery and shade house space is needed and labour, and eventual plant outs to preserve lineages long term and to set up resistance screening arrays will take a lot of ground and work at multiple sites because you don't put all your eggs in one basket. The array at the right there is not native guava. That's a trial layout of 900 Melaleuca quinquenervia, another species affected by Myrtle Rust, although not quite as drastically. But that's just to show the scale of what a plant out of 900 looks like. And in the next slide, we also see similar arrays for resistance trials under field conditions of lemon myrtle that have been set up at a couple of sites, one in New South Wales, one in Queensland. So as you can see, a fair bit of ground and a fair bit of labour involved. Now, I mentioned that we don't want to put all our eggs in one basket, so we're dispersing collections of native guava and eventually of other species as well to multiple sites of collaborating institutions, botanic gardens, potentially universities and others, in order that a single event, be it bushfire, major frost, whatever, doesn't wipe everything out, and that we have a replication ability for to replace lost lineages at any one site as well. But there's a price to be paid for that kind of dispersed collection, and it means more space and more work for those recipient sites. They're not cost-free for those sites or for the Central Propagating Authority. And there's labour time involved in particular, which is very scarce, as we know, and there are some dollar costs, although those are not huge by and large. But the plant out and the frequent monitoring that is necessary, both for Myrtle Rust and for other problem pests and so forth, and spray treatment periodically if there is an outbreak, those do cost something. They cost time, they cost money, and there are also associated hazards with fungicide use, and of course there are public perception issues. And then there's the records management task as well, which for small regional botanic gardens can be quite a large proportion of the effort that goes in. So an ability to assist particularly the smaller operations that are collaborating with this dispersed collection would be a good thing. We're guided in all this by towards the meta collection policy that was, or not policy, but a guidelines document that was developed by Botanic Gardens Conservation International some years ago, and we're attempting to ensure that we operate in good practice. Now, there's a couple of clusters of collaboration that are emerging around this dispersed collection concept in the Myrtle Rust space at the moment. The most developed one is in the southeast of Australia, shown in green on this slide, and that's largely been around native guava up to this point, although we're hoping to utilise the same network to some degree to disperse a couple of other species as well. But we've got to be careful not to saturate the ability of recipient gardens, obviously, to host material. Then in northeastern Australia, shown in red, there's a focus of activity organisationally centred at the Australian Tropical Herbarium in Cairns, but with the ex situ work largely happening at JCU, James Cook University, Townsville, where Brandon Espy has succeeded in capturing a considerable number of lineages of some of the northeast Queensland species that are imperilled by Myrtle Rust. So there's, just for this one species, there's quite an array in the southeast of collaboration involved and coordination, and making sure that record keeping works over a span of different databases across those gardens and all that sort of thing. Fair bit of work. In Queensland, I just want to note the work that's being done at the DAF, Department of Agriculture and Fisheries Forestry Nursery in Gympie by Tracy Menzies and others in propagating native guava there from Queensland provenances. And in northeast Queensland, as I mentioned, Brandon Espy at James Cook Uni has been capturing species for some time now. And he's got living samples of over 36 Myrtle Rust impacted species so far. That's not to say they're fully adequate collections in terms of where we want to be, but it's a huge start, and all power to him. So all that effort around Myrtle Rust, we've got perhaps 5 of the subtropical species now that for which we have adequately representative collections for long-term ex situ conservation and development work, and perhaps adequate collections for 2 or 3 in the tropics. It might be slightly more by now. I haven't checked in with Brandon recently. And for only 1 species do we yet have the critical lineages fully dispersed for security under the meta collections model. That's native guava. We're working on it for a couple of others. So for Myrtle Rust, we've only got 40 or 45 species to go. What's the problem? But that's before screening and before breeding and before rewilding, and it still hasn't got to southwest WA. So how unique is this Myrtle Rust generated demand for ex situ space and facilities and labour? Is it just a Myrtle Rust blip on the landscape? Well, this slide is borrowed from Darren Crane. Thank you, Darren. And Darren's adamant about pointing out to as many people as will sit still to listen that the wet tropics World Heritage Area of Australia is the 6th most irreplaceable of all the protected areas worldwide, 173,000 of them. It's the 6th, and it's the 8th on the basis of threatened species. And it's the 2nd most irreplaceable natural World Heritage Site, and there are some 80 plus species of vascular plants endemic to the cloud forests of northeast Queensland. And for those cloud forest species, climate change is an existential risk that's already in operation, and a direct risk, not just a sort of aggravator of other existing risks. Rainfall and cloud cover are already diminishing on those peaks. Plants are moving uphill, and there's nowhere for these cloud forest species to go. So the conservation response to that is the TROMPS program, the Tropical Mountain Plant Science Conservation Program, which is a collaboration of multiple institutions, but largely coordinated out of Australian Tropical Herbarium in Cairns. And their concept diagram looks like this. and there's a great deal in common there with the Myrtle Rust schematic. The contribution of the seed banking research, the propagation, the genetic analysis, and that big blob in the middle at the base of the ex situ distributed collection with all the associated activity around that. And if you look at the two schematics side by side, there is, as I say, a lot in common between the two, and they are drawing on the same resources and the same kind of concepts and facilities. So the TROMPS collaboration at present looks something like this. They've got a cluster in North Queensland, but they've got a number of southeastern Australian and one New Zealand collaborating partner. And some of the Southeast Australian partners, of course, are the same institutions as are helping with the Myrtle Rust meta collection. So how many species are involved in the TROMPS program? Well, they've currently got about 33 of these cloud forest vascular plant species in ex situ conservation, although only some of them are yet at a fully representative level. And that's out of an estimated 80 or 90 vascular plant taxa that are endemic to those montane cloud forests. So that leaves sort of 50 plus that are in scope for the TROMPS program, but are yet to be added to this meta collection and for which funding as yet has not been allocated. So that's, 40, 50 in the Myrtle Rust space, 50 plus in the TROMPS space. And then on top of that, there's an rapidly increasing frequency of ordinary ex situ and translocation actions related to conservation that botanic gardens and a few other institutions are already undertaking that aren't connected with the mountaintop situation or the Myrtle Rust. Now, there have been a number of partial reviews of translocation actions and the ex situ dimensions of that in recent years, but the first real overall review of translocation in Australia is that by Silcock et al. In 2019. And Silcock et al. Reviewed just over 1,000 translocations that have occurred since 1976, involving 276 different taxa. About 70% of those were conservation translocations, the sort that we're used to in the Botanic Gardens situation, and about 30% were mitigation translocations to satisfy development conditions and things of that sort, largely conducted by contractors, although there's overlap between these two. One really noticeable trend is that over 85% of all those translocations that were documented have occurred since the year 2000, and over half of them since 2010. And that's true in the mitigation area as well, with 30% of the total occurring just in the past 5 years, up to 2019. A further thing that's kind of a good trend, but also a worrying one, is that the number of plant units being used in translocations has averaged less than 50 in the past. But there is good data worldwide that shows that the number of founder propagules, founder units that are used, there's a direct correlation between a higher number of those and a higher likelihood of successful establishment, which is your goal, of course, with translocation. So the trend in the future is likely to be for more units, hence more space demands and labour demands on nurseries and on maintenance facilities, shade houses and so forth. And the prevalence and imperatives for translocations will continue to grow for southeastern Australia, and I would suggest probably northeastern tropical Australia as well, under projected climate scenarios. The takeaway from all this is that the demand trend for translocations will continue to increase for those of us in the southeast, and the scale, the number of plant units involved, is also likely to increase the demand for that. Although genetic optimisation may provide some partial counter to this, although at a cost of losing some allelic diversity along the way. Now, Western Australia has a rather different trajectory and a different bottleneck problem. Their main problem in the southwest at the moment is trying to find recipient sites that are phytophthora free. So their bottleneck situation is rather different. They've also had translocations built into more plants for a longer period of time. Won't dwell on that. So we have a strong upward trend for ex situ conservation actions in general, both seed and tissue banking, standing ex situ collections of whole plants, and translocation. Now, there's no national figures to hand that I've been able to locate, but I would suggest that the trend in New South Wales probably holds good for South East Queensland, maybe North East Queensland over time, and probably Victoria. We do need to ground truth at that. I'm indebted to Chantal Doyle, who's allowed me to quote from a manuscript that's currently in review. As of financial year 22-23, there were 897 plant species listed under the New South Wales Biodiversity Conservation Act, and 30% of those, 269, 30%, have been assigned translocation or ex situ based actions by the Saving Our Species program, the state threatened species program. 136 of those are earmarked for translocations actions, which necessarily involve an ex situ phase, and 172 for other forms of ex situ conservation, largely seed banking, but some for tissue banking as well. Relatively few of them have yet been investigated for genetic structure to enable the best management of meta collections and actions. And of course, there's seed biology and other forms of research that are necessary for effective management as well. And Chantal and her colleagues note that the advent of genetics and maternal line collections has meant increased labour and resource requirements to process, maintain and store these ex situ collections. So our capability and priority pressures on ex situ collections in botanic gardens and in a few other similar or parallel institutions include increasingly expensive fieldwork, increasingly complex and time-consuming accessioning, to good end, but increasingly complex. The competing priorities under the seed and tissue banking area between ordinary species, all species goals, threatened species priorities, and then thematic or funding dictated priorities that we may have. And there are particular pressures in the propagation and maintenance area, including space and skilled horticultural and scientific labour, and the crossover between the two, which I would suggest is increasingly important and is under-recognised. There's also potential pressures in the availability of genetics and seed biology areas, but that's less pressing in some ways. ASBP, of course, has its member institutions. This is a slide put together by Amelia some years ago, but there is no way that these institutions alone can meet the need. We do, of course, have 130 plus BGANS members. Strangely enough, there's no map of that available that I've been able to determine, but their capabilities for participating in conservation activities vary widely between different regional botanic gardens. They're all willing, they all want to participate in conservation activity, but having the capability to do so for some of these projects, whether it's a climatic situation or a skills base or facilities base or whatever, isn't automatic. So there's issues around what conservation can be done in different places. In the BGAN survey last year, the question was asked of a relatively small number of respondents, are you funded for additional taxa? A majority said no, and I would suggest that that holds good for the non-respondents as well. Getting back to Myrtle Rust, and I'll close with this slide because I'm way over time. We brought Richard Sniezko from the US Department of Agriculture Forestry Service out for the Australasian Myrtle Rust Conference last year. Richard works and largely runs the Dorena Genetic Resource Centre in Oregon, USA, and they specialise in tree breeding, particularly for disease resistance, but for some other purposes as well, for conservation goals and also for timber production goals. It's a dedicated unit that just specialises in that for tree species. And many of our Myrtle Rust species are, of course, trees, not just shrubs. And many of the tropical mountaintop species are trees or large shrubs, not just small shrubs or herbs. And I would suggest that we really need to be having a very close look at the Dorina Genetic Resource Centre, its facilities, and potentially as a model for Australia as well. Richard advances a number of prerequisites that he suggests are necessary. We've got the strong core group passionate about the affected species. The problem is to infect our upper levels of our food chains in the bureaucracies and the political world with that same passion and that same sense of urgency. We need a review of other successful resistance programs. This being Australia, we do things 9 different ways if we possibly can, and that's not a good thing. Any program along these lines needs proper structuring and staffing and a continuity of funding and personnel, which is also completely contra our current culture outside a few long-term institutions. And we need a series of 5-year plans and reviews, and I don't think that would be a problem. We also need to foster a dedicated cadre of technical staff in science and horticulture, and as I mentioned, the crossover area between the two, and value the skills at that level rather more than we do now. Richard's key piece of advice, and I really take this one on board, is that research, while it's absolutely necessary around all these species and justifiable and fundable in its own right from other sources, for a facility of this sort, whether it's a single centralised facility or a dispersed one based in existing institutions, the research should service the resistance breeding goal. If disease resistance is your conservation driver, research should not be itself the main driver. Output of resistant genotypes is the goal, and everything else should be subordinated to that. That's a clear lesson from the history of Dorina, and one which I think we should examine closely. And finally, Richard proposes that we really need to get our act together and produce a prospectus of costed options. We sort of know what we could do with $200,000 already. That's what we've been doing since 2019, little bits of piecemeal funding of that sort of order. Really significant, actually, and we've been able to deploy it to good effect, but not sufficient for the long-term nature of recovery that these species demand. So what will we do for $2 million? What would we do with $20 million? And importantly, would we do it separately or would we do it together? Thanks very much.
Thanks, Bob. That was like an amazing, I can't believe you fitted so much in 25 minutes. Brad, how are we going with questions there? The key sort of messages I took away from that, Bob, was across the number of scales you spoke about, from that labour and capacity at that, botanic gardens level for your diffuse collection, the cooperative work we need to do to work together across agencies, just as sort of at that kind of, mid-level, and then the need for that national coordination and investment support to make this all happen. And I mean, the important thing is we do have the action plan to help frame what we need to do, and presumably your prospectus of costed options that we've left here on the screen would pick up, the pathways that you've identified in that action plan. So yeah, let's have a look and see.
I'll just comment briefly on that, Kathy. The Myrtle Rust National Action Plan was first formulated in 2018 at a point where we were trying to get even rudimentary political and bureaucratic attention to the Myrtle Rust problem, and it tried to address a range of issues related to native plant conservation, not so much the production situation, but that included research goals and things of that sort, as well as the kind of recovery action goals that I've been talking about today. So the National Action Plan, even in the revised form of 2020, covers a broader gamut of things than what I've been talking about, as it should do. But it's worth noting that while it's increasingly providing a bit of an operational framework in New South Wales and Queensland and nominally for the Commonwealth, it still isn't adopted or funded in any formal sense. The development of a program around these recovery actions has been largely happening at state level in New South Wales and Queensland at the moment, which is good because they're running with it. But we need to formulate either an expanded version of the National Action Plan or a subset of that, a document sort of sitting under it, nested under it, that does address some of these issues as well. Once we have a consensus in the area of what we need to do and what it's possible for us to do given the disparate nature of our jurisdictions and institutions.
So it'd be worthwhile you just talking a little bit about the fact there is a working group that operates from time to time, Bob, and that, you know, to some extent we need to, I suppose, put more weight and effort around the work of that group. You've gone on to mute, Bob.
Beg your pardon. There is indeed a National Myrtle Rust working group. This came under Commonwealth convenership a couple of years ago. It had previously been an informal and unresourced group operated by the sort of circle of concern around Myrtle Rust, which did quite a lot during that informal period to advance the issue. The current Commonwealth convened committee meets a couple of times a year and is a bit of a clearinghouse of information, although a great deal of what goes on happens out of session, and it's not really a decision-making body as such, but it is between conferences, it's the largest meeting point we've got at this point. So yes, it's important that we use it, but we shouldn't expect it to be the vehicle for collaboration development and things of that sort.
So who funds Dorina over in the US? Is that a combination of, you know, state and private funding?
It's primarily funded by the US Department of Agriculture. They do have, I think, some private funding. I don't know the details of that yet. But as I mentioned, a close look at how they operate, including that funding base, I think is something we should undertake.
Okay. Now, Brad, have we got any questions in the chat or the Q&A? Published. Sorry, everyone, on the troglodyte on the Teams. Okay, so let's have a look. Just go and flick down here. Okay, from Nico. How do we mitigate the costs in those collections you refer to? Right, sure.
Well, and for the costs for the recipient institutions, in the case of native guava, as far as it's gone at the moment, we built into the original grant application, which we succeeded in getting in 2021, a relatively low modicum of funding for the recipient gardens to assist with site selection, site preparation, and the actual installation and monitoring costs for the duration of that grant, which finished in basically with an extension, it finished this year. So that does nothing in terms of long-term sustaining of these matter collections. There's been so much goodwill, I've got to say, around these collaborations, as there is with many of the conservation collaborations that we undertake in the botanic garden sector, that a great deal has been leveraged out of the situation on a pretty much a shoestring of money. And I'm sure that will continue to be the case, but there's limits to what people can accommodate without support. So building sustaining money for the recipient institutions into all future grants around this area is an obvious mechanism. But if we were to advance a prospectus proposal for either one big facility or a dispersed facility with the large nodes in existing gardens, whatever it might be, a recognition in that proposal of the potential role of regional botanic gardens and other small collaborators, and a financial allocation for their support of that whole show would be an appropriate thing to do.
Yeah, the whole recurrent funding around curation applies across the board from seed banks through to our living collections. Okay, so Brad's just asked a question in relation to myrtle rust susceptibility of the endemic Myrtaceae in WA. Are you aware of any work to test in that?
There's been some testing of WA species off and on since myrtle rust first arrived in 2010-11, initially by Louise Moran at CSIRO, more recently by various people working in and with the Plant Breeding Institute at the University of Sydney, and I think some in Queensland as well, not sure about that. There were recently 15, I think, WA taxa tested. They were all capable of sustaining myrtle rust, but that's true of Myrtaceae in general. These things are inoculated under lab or glasshouse conditions favourable to the rust because you really want to see what it can do. And out of all those that have been tested so far, a study in 2018 identified that only 3% of the tested Myrtaceae had failed to become infected under those highly favourable conditions.
Wow.
So in a way that tells you everything that you want to know, and in a way it tells you nothing about what you want to know, because it's not a predictor of what will happen in the field. That said, in the southwest of WA, in the moistest regions of southwest WA, there's over 1,000 Myrtaceae species, and a significant number that have been exposed in horticulture in eastern Australia do contract myrtle rust. While we don't yet know quite how myrtle rust will behave in a Mediterranean climate like that, that southwest area is a zone of extreme concern, and the West Australians are certainly treating it that way. They're doing preemptive seed collection. They have an advantage, of course, in the much greater proportion of their Myrtaceae is orthodox seeded and can be stored as seed compared to ours.
Yeah. Okay, let's just scroll up here. So Brad's also asked a question about what we can do as individuals to help in combating myrtle rust. And I noticed on the slide of Richard's ideas is the whole idea about rust busters, which we've had for other sort of invasive and exotic species.
Yeah. And advocacy and awareness activities themselves need paid labour input to be really effective, as we all know.
Yep.
While you can achieve a certain amount by everybody going off in their own directions and beating drums in discord, it's a hell of a lot more effective if they're in unison. And simply finding the resources to do that has been a problem. ANPC has played a significant role in that, I think, but we could, I think, be looking at the possibility of entering a new stage of advocacy. Again, once we have consensus about how we implement the general conservation strategy that's been outlined in the National Action Plan and has been pioneered in relation to this handful of southeastern species. Once we have consensus around that, I think we could be looking at the possibility for something like a Myrtle Rust Foundation to try and garner private and philanthropic funds at a much greater level than we have so far, in conjunction with government funds. Neither of them is going to do the job alone, but something like a foundation approach with crossover, I think, does have possibilities. Again, like the Dorina idea, it's something that we need to study very carefully, and that would itself take a year or two of very careful paid preparation work to launch properly. Not something you do ad hoc.
Yeah, I mean, we certainly know we've got the tech to enable it to happen too. We want to kind of, leverage that community citizen science element. So from Karina, how do you recommend scientists to better partner with the horticulturalists and restoration practitioners, which you raised a couple of times in your talk? And Karina said she's found it difficult to recommend genetic optimization for genetic-based resistance and diversity. She means in working with the horticulturalists.
Right. I imagine she means by that they're not seeing the need or finding it difficult to uptake within their existing priorities. Is that the gist?
I'd say so,
yeah.
Unless Karina wants to add to her question online.
Well, taking the first element of the question, then how do we assist that horticultural head knowledge to be exchanged and developed and so forth? There's pretty strong siloing in some of our institutions between the scientific and the horticultural staff, and things that help break that down, like cross briefings, cross familiarisation, really trying to understand the competing priorities that may exist with your project and why things aren't getting done as fast as you'd like and why the bloody scientists don't get their act together. That kind of breaking down of silos is something that individuals can begin to do themselves. But also so is encouraging horticultural staff to think about writing up their head knowledge. And while publication avenues for that are rather few in Australia compared to overseas or some overseas countries, encouraging horticultural staff to publish more, either solo or in conjunction with scientific or other staff or other externals for that matter, would I think be a valuable sort of thing. It would also help improve the recognition of the horticultural skills as an integral part of the conservation effort, of the ex situ conservation effort.
I understood as part of your AMPC project, you had a bit of a community of practice set up to actually sort of bring the practitioners and the scientists together as well. So expanding that model potentially is an option.
Potentially, and BCARM, of course, has taken an initiative along those lines with its periodic forums that Veronica Viola initiated, which are deliberately not structured as webinars like this one. They're not recorded. They're simply an exchange of information opportunity where people can not simply speak their mind, but they can tease things out at a very practical level as well. And having that running in parallel with more formal communications channels, I think, has been a very good thing.
So is there an opportunity to open that network to a broader range of practitioners, Bob? I mean, I know it was very focused on the localities that you were working.
There is, and in a recent funding application to the Commonwealth, an expanded set of consortium members for a slightly expanded bracket of species was proposed. We, inverted commas, succeeded with that funding application. We got half the amount of money that had been applied for and that was necessary. And we're in the process now of recruiting a program coordinator and adjusting the project plan accordingly. So yes, expansion is possible, but doing it on an ad hoc project by project, consortium by consortium approach continues the piecemeal approach that we've been taking to the whole problem of both Myrtle Rust and Tromps as well. It doesn't really set us up with ongoing capabilities.
Yeah, ditto. The whole capacity building thing, it's really hard to get funding and resources in that space at the moment. Doesn't matter which area of conservation you're talking about. So I've got a question from Gareth Holmes. if the Myrtle Rust extinction rust is so critical for some species, would you consider a more focused genetic introduction of resistance genes if they can be located
Hi, Gareth. I
in
assume,
some remaining genotypes? A CRISPR approach may be suitable.
yeah. Look, that has been looked at overseas, and it's something that I canvassed fairly briefly in a review that I wrote of Myrtle Rust impacts back in 2018. It's an approach that is being looked at for some species overseas, and of course it's being used in animals quite a bit, but not so far in plants outside the agricultural purview, and certainly not for woody plants, with a very few exceptions. They are looking at it, I think, for North American chestnut as one of the possibilities for recovery of that species, formerly a community dominant over much of eastern North America, but reduced to virtually nothing now. It's more interventionist yet, more interventionist still than what we're proposing. And for species that truly have little or nothing in the way of tolerance genes or traits for Myrtle Rust, it is something we should look at the possibility of, I agree, but it's not something that we should advance lightly as a solution. There's a long way we can go with the ordinary classical breeding kind of approach. That's more palatable for people. It doesn't arouse the whole genetic modification sort of issue in quite the same way, although I think we could sustain an argument that even GM is better than extinction. But I don't think it's something that we should be canvassing as the main thrust at this point myself, but we should, as a background to everything else we're doing, be keeping a very close eye on the possibilities in that direction, because it may come to that for some species down the track. But the main thing, the main problem at this stage is keeping our options open, and that involves germplasm capture and maintenance. That's the fundamental problem. That's the point we're at the moment.
And then we don't have to triage down the track. Okay. Is there any more questions, Brett? I can't see any more in here. No, I've got the thumbs down. Bob, that was really lots of food for thought. I think you've captured the immediate priorities, and then some of this work that ideally we can do if we can get that sort of coalition of the willing, as you say, that consensus. I'd really like to think that we can present a little bit more on the work that you've done across the East Coast to actually show people how effective that was and the range of partnerships that were actually involved in that project. Any of you that are attending the ANPC conference in Toowoomba, I'm sure that there's going to be quite a few sessions around Myrtle Rust. Something that Bob didn't mention, which is critical, is that in Queensland in particular, we actually don't have an actively curated conservation seed bank at the moment. And given the likely impact of Myrtle Rust across that state, that puts them at a bit of a disadvantage. And you obviously saw the work that's going on in the tropical parts of Queensland. We haven't got the same sort of focus and effort and support in southeast Queensland. That's happening through Jeff Pegg and the DAF, but on the conservation side, that's obviously an obvious gap. Just double check, no more questions before we head off. Bob's presentation will be presented as a video file, so if you've got colleagues that weren't able to attend today, please share the link. We will provide that post the webinar. Our next webinar will be capturing some of the issues that haven't got a lot of coverage over in the East Coast, which was the impact of the severe droughts in WA and the effect that's had on their shrublands and their very important forests. And there's also a little bit of research from Tasmania around the same sort of drought-related climate impacts on plants from those recent sort of, sustained drought events. So stay in the loop for that. We will obviously, circulate internally and externally through our networks. And I'd just like to thank everyone for staying online as well for the full hour, because I think those conversations at the end, Bob, and the questions are always, really insightful to see what people are interested in and how they can get involved. So thank you, everyone, and a big thanks to Bob, our Czar of Myrtle Rust, who I think sort of presented an amazing pathway forward. And I think we've got a national working group, Myrtle Rust working group meeting in the next month, and that'll be a place that we can sort of carry on some of these conversations and look at some of the priorities. So thank you all, and particular thanks to you, Bob.